Hey there, cucumbers. Fluorescence microscopes are a pretty interesting piece of equipment used in the lab. The most useful feature being that they're able to illustrate specific components of cells using endogenously expressed proteins with fluorescent tags such as GFP or with fluorescently labeled antibodies after fixation. Going over the components of this setup, first you have the camera and laser modules. In this case, it's an epifluorescent setup, which means that the laser illuminates the sample through the objective lens, rather than a fluorescent setup where the laser and objective are positioned on opposite sides of the sample being imaged. As far as I can tell, epifluorescent setups are more common. Then you have the control wheel for positioning the sample in X, Y, and Z, the sample holder and objectives, and finally the light source that allows the microscope to be used for standard light field microscopy as well. After the hardware, there's also the software to consider. This is where you can change settings such as camera exposure and gain, the illumination settings, change the objective, take multiple images at different focus levels to create a 3D representation or Z-stack of cells. You can change the laser excitation wavelength and view the captured images. There's a lot more features as well, but those are likely the most common ones that you'll be required to use. With all that background out of the way, I thought I'd give a quick walkthrough of the process involved in actually capturing a Z-stack of a cell from start to finish using all four laser channels available. Since I'm using a 63x oil objective, I have to add a drop of glycerol onto the objective first. Then I can add my slide with my samples on top. The top front wheel controls forward or backwards. The bottom front wheel controls left and right. The top back wheel controls up and down or focus. And finally, I can use the control wheel to adjust the sample position to whatever cell I want to take an image of. One interesting feature is that you can actually see the excitation laser colors change as you change the selection in the software. In this case, the blue laser corresponds with the green output channel, the green laser corresponds with the red output channel, the apparently absent laser is UV, which isn't visible to the naked eye and corresponds to the blue output channel, and the red laser corresponds with the far red or pink output channel. Each emission color corresponds to a slightly higher energy excitation color. That's a basic feature of fluorescence, and I think it's kind of cool that you can actually see it working here. I also wanted to try to show what the cells look like through the eyepiece of the microscope instead of from the camera and computer. There's actually quite a big difference between the two. The eyepiece has a much larger field of view, and because your eyes have such good dynamic range, you can move the sample without any motion blur, which isn't the case if you have longer exposures on the camera. The video I captured really doesn't do it justice, so you'll just have to trust me on that. To perform the Z-stack, all you have to do is move the height control wheel in one direction until the cells are out of focus. Mark this as the beginning of the Z-stack. Then move the control wheel in the other direction past the in-focus point until the image is out of focus again. Mark this as the end of the Z-stack. Then you can hit start, and it should acquire the Z-stacks of all the channels listed. Fairly straightforward, but I think watching the images get captured is also quite beautiful too. And that's it for the fluorescence microscope video. Thanks for watching. If you enjoyed it, don't forget to like, comment, and subscribe.